slide, but it's important for me to review the slide of an antibody because we're going to talk about the class of antibodies, and they really depend on the FC. So basically, the antibody is basically a secreted what? BCR. B cell receptor. Remember, a B cell has that Y-shaped BCR, right? And as long as that BCR is in the membrane, it's called a BCR. Now, when an epitope binds the what? Variable region, this is my Y, right? This is your BCR, right? Looks like a Y. Okay. The arms have hands, right? The hands are the variable regions. They actually bind to a specific what? Epitope, right? Now at that point, that B cell that binds some epitope, it remains a B cell. But it's going to be activated, which means what? It's going to make clones of itself. All the clones have the same what? Y, makes sense? All the clones have the same what? Variable regions, which means they recognize the same what? Epitope, correct? Why am I going slow and going into details? This chapter is not intuitive, right? It's not intuitive. You have to explain how things work. You can't memorize nothing. I'm telling you straight up. You have to understand if this binds that, what happens after that. You have to, okay? If you want to pass this chapter, I'm being frank. Here we go. Now, a clone of a B cell, right? All the clones of the B cell, they are now plasma cells because plasma cells, they actually do what? They actually secrete their Ys. Those Ys that are secreted are your antibodies. Make sense? Got it. Now, I talked about this stem, right? That stem is usually in the membrane. Make sense? If it's a what? BCR, correct? But it's now floating in your blood. That stem is called the FC region. Why am I spending time on this? It's not trivial. Because you have 5 different types of antibodies, and depending on if it's IgA or IgM or IgE or IgD, depends on probably, I don't know, that thing right there, correct? So that's why I spent time on that. That FC region changes. And depending on that FC region, I'll get into it, depending on that FC region tells you whether you're IgA or IgE. Makes sense? So with that said, let's go on to here. I believe we left off here, correct? Okay, great. Now, I just mentioned, okay, there are 5 different classes of antibodies. It depends on the type of antigen they are encountered, the port of entry, is it the respiratory tract or the UI tract or the reproductive tract? How are they getting in? They being antigens, okay? Also, the type of antibody function needed. Remember last week we talked about different ways antibodies can work. I will know all those ways, right? Whether it's agglutination, opsonization, neutralization, all that stuff. Know that, okay? Those are ways antibodies can attack and take out what? Antigens. Right? Now here are your 5 classes of antibodies, okay? They are based, I just said it right, they are based on that FC region. So whether it's IgA or IgD, it's based on the stem. These FC regions are here. So all these guys here, whether it's mu, gamma, alpha, epsilon, delta, these are your types of FCs, got it? And that will tell you your class of antibody. Let's get into that real quick. Now you're like, wait a minute, I don't see that in my notes. I know you don't see it in your notes. That's why I put it in red. So it's an indicator that it's not in your notes, but it's going to help you. Makes sense. It's going to help you. Because I know some of you like words, so I got words for you. I don't really like words. So here we go. Bless you. IgM. Okay. Basically, it has a mu FC. It's real easy, right? So basically, IgM is an antibody that has the mu FC. It's real simple, okay? IgM, what you need to know, that is the first. I mean, it's the first antibody that you will ever secrete. I like this. Pictures. Great. Take it. I don't care. Do whatever you got to do. I'll even smile for the camera. Okay? IgM is the first antibody ever made. Whenever your B cells, I repeat, whenever your B cells encounter some epitope, the first antibody that will be made and secreted will always be IgM. Okay? Now, IgM, it can act as a BCR. When you see that, what does that really mean, okay? It means it can remain in the membrane. It should click, right? If I say BCR, it's a Y-shaped guy in the membrane. It can stay there, or it can be secreted. Now again, if it's secreted, it's now your antibody. Here's the deal. Secreted IgM forms a pentamer. Penta means 5. So in other words, if you actually secrete out the IgM, 5 of them get together and they just come together and they form one single molecule with 5 antibodies on it. So far so good, it forms a pentamer. Now, what can IgM do, okay? It can activate the complement system, right? MAC attack, inflammatory response, agglutination, or neutralization, okay? Those are the functions of IgM. Next is IgG. So again, all antibodies start off as IgM, and then they can do something called class switching. Now, class switching is basically when they turn into another antibody, simply put. All they have to do is do what? Switch out their Fc, right? If they change your Fc, they literally become a new antibody. Make sense? As far so good? Now, IgM can become IgG. Why? Fc is now gamma. Now, IgG is by far the most common antibody. It accounts for about 80% of all your antibodies that are floating around your body is IgG, and it's the longest lasting guy. Real quick, I'm not going to go into all of that, but here's all the things IgG can do. What is ADCC, right? Antibody-dependent cellular cytotoxicity. So go back to your notes to figure out what that's all about. That was Tuesday's lecture, or Thursday of last week. Go back to your notes. Okay? But really important here, IgG can actually cross the placenta, meaning it can actually protect the fetus in the womb, okay? So IgG can actually cross the placenta. Got it? Real good. Three more, real quick. IgA probably has the alpha Fc, okay? IgA is primarily found in bodily secretions like saliva or milk, for example. So I just mentioned it. So IgA is secreted in milk. So if you're breastfeeding, it's really good because that's how newborns get some kind of protection. They get IgA in the breast milk. So it's really important. Bless you. Now, secreted IgA, if it's found in bodily secretions, it forms a dimer. So it's secretory IgA. So if it's found in a bodily secretion, it's a dimer. So two of them come together, and they stick together, and they act to do either neutralization or agglutination. But the key thing is to really know it helps protect your newborn. It's found in breast milk. IgE. Now, IgE is really peculiar because it really doesn't act like an antibody. It really acts like a signal molecule. It signals other cells to do something, okay? So, for example, IgE, it can bind to eosinophils. I think we talked about them last chapter. And if they bind onto eosinophils, they cause those guys to attack worms. Also, IgE can trigger allergies. In other words, if you're like me, you have lots of hay fever, you probably have lots of IgE floating around your body. In other words, IgE can also bind to mast cells and basophil cells and tell them to secrete histamine, which causes all kind of havoc in your body. You take Benadryl, okay, you know that story. No. IgD, okay, that one, we don't really know the function because not all mammals have IgD. And if they don't have it, there's no ill effects with that. So in other words, you don't need it. We don't really know what it does anyway. But you should know it is a class of antibody, and it has the delta Fc. Make sense? So we don't know its function, and not all mammals have it anyway. So we don't really care about it, okay? It does something, we don't know what it is. Oh, no. This table summarizes two things I mentioned. Do you have to know everything? No. But I highlighted one thing you should know, right? What does it do, right? What can it do for you? Now, I would know kind of what they look like. I mentioned two of them, right? I said IgA. If it's secretory, meaning it's found in a fluid, it's going to form a dimer, right? Which means you actually have 1, 2, 3, 4 variable regions, right? They have to bind onto an epitope. And I did mention, right, IgM. I said if it's secreted out, I said it forms what? A pentamer, and you should know that. In other words, IgM can be still found in the membrane. It could be a BCR, makes sense? But if it leaves that membrane, it forms a pentamer, got it? So that you should also know. The other guys is regular Ys, okay? They're all regular. Now, real quick, T cells or T lymphocytes, we already said they're involved in what? Cell-mediated adaptive immunity. They do not secrete antibodies, okay? They primarily target intracellular parasites or even cancer cells. That's their job. We know they're born here, right? But they're called T cells because they mature where? In the thymus. Real easy. They can be circulating in the blood and the lymph. So they can be kind of going around looking for things to attack, okay? They can also be found in the Peyer's patches. Remember, they're found in the intestines. Now, I put an arrow right here because this point here really goes here, and that point really goes there. That's what this arrow is telling you, because it makes sense to say T cells have their own receptors, right? Just like B cells have their BCRs, T cells have their own receptors, probably called TCRs, right? T cell receptor. They all have them. And then, if you say that, then this makes sense, because on their TCRs, they also have what? Little hands. Look a little different, right? They have hands too, and they bind very specific epitopes. Make sense? So move this point up. So T cells have their own receptors, and the receptors have their own hands, right? Looking for something very specific. Make sense?
The Peyer's patches they're part of the moth, right?
Yes. So Peyer's patches, they're part of the moth. Absolutely. Absolutely. Very good. Now, real quick, here's a picture of a TCR. It doesn't look like a Y, right? It's not a Y shape at all. It just looks like a little, I don't know, maybe an L or something. But what you should know is two things, okay? A is a glycoprotein. Did you see, these guys are sugars, okay? Those are sugars. Did you see any sugars on those BCRs? No. There were no sugars. If they were, I would have mentioned it. Did I mention sugars on those?
No.
No. But I'm mentioning with the TCRs, they have sugars on them, so they're called glycoproteins, right? Glyco means sugar. Got it? So the orange are proteins, the green guys are sugars, they're glycoproteins, and on top is basically their one hand. So TCRs have one hand, right? BCRs have two hands. Hands basically recognize and bind a very specific what? Epitope, right? Now here's the deal, I'll tell you right now. They don't work like a BCR. A BCR can literally attach to an epitope, right? It can do that. They can't do that, okay? They can't. This is here. They do not recognize epitopes directly. So even if there's some strange pathogen in your body, these T cells cannot bind that pathogen directly. Unlike a B cell can. B cells can do that. T cells cannot. What's happening here is that TCRs, they can only bind some epitope when that epitope is bound to this thing called MHC. Now, MHC stands for Major Histocompatibility Complex, or just MHC for short. Now, in subsequent slides, I will tell you exactly what an MHC is and what it does, but right now, the only thing you should understand is that a TCR cannot directly bind an epitope. Right? BCRs can, TCRs cannot. TCRs bind epitopes when the epitopes are bound to an MHC. Simply put, MHCs present epitopes. That's their job, right? That's all they do. MHCs present some epitope, and then you have a T cell come along with his little TCR, and they do that basically. And we'll get into what happens when that happens, right? Again, no memorization here. You really have to understand how things work, right? Where's the clicker? Oh, here it is. Got too excited. Blah, 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 blah. I mentioned this, right? Basically, these T cells target what? Intracellular pathogens like viruses, okay? Intracellular worms, intracellular bacteria, anything inside the cell, T cells will target it. Now, there are three basic types of T cells. They're dependent on what they do, right? Their actual function, but more than that, the types are dependent on what kind of glycoproteins they have on their membrane. That's why I mentioned to you, right, that TCRs have sugars on them, right? So depending on what kind of glycoprotein they have, and their function tells you what kind of T cell I have, okay? Let's do that real quick. Cytotoxic T cell, or TC. So in other words, in this lecture, TC is a cytotoxic T cell, or basically a CD8. I would know both, okay? Because you are going to be healthcare professionals, you should understand that. You should. CD8 is.
What does CD8 stand for?
It stands for cellular differentiation. This talks about the type of glycoprotein on the TCR. Now, the number is just the order they were discovered. That's simply put. So their number is the order they were discovered. That's all. But you should know, okay, that a cytotoxic T cell is also a CD8, because you may be doing some T cell counts in a patient, and they're not going to say TC, they'll say CD8 versus CD4, right? You should know what they stand for. Okay? Now, they are cytotoxic. Cyto means cell, toxic means kill. They kill cells. So these are the T cells that actually take out cells that are infected with the virus or cells that are cancerous, okay? They take them out. They take them out. Okay? They kill, literally. Now, the commander of the whole army is that guy, the T helper one, or CD4, okay? CD4 are the T helper cells. I'll tell you right now, there are two types. We'll get into it later on. There's T helper 1 and T helper 2. They do different things, so save that for later. I know Debbie's like, tell me now, I'm going to tease you a little bit. There are two types of helper cells, TH helper 1, TH helper 2. They're both known as CD4. Now, they regulate, this is why they're the commanders, right? They regulate the activities of the other immune cells. In other words, they regulate either the TC cells or they regulate the B cells. So here's the teaser, Debbie. The T helper 1 guys, the type 1 helpers, type 1, they regulate the TC guys, TC being who? The killers, right? They regulate them and macrophages. All this is important. The T helper 2 guys, I told you there are two types, right? The T helper 2 guys, they regulate B cells. Now already you should understand, if you take out the helpers while your immune system does not work, I just told you they regulate either these guys who actually kill cells or the B cells that actually do what? Make antibodies. So if you take out the T helper guys, your immune system doesn't work very well. Make sense? Why am I telling you this? Because, say it. Because HIV targets CD4 cells. It takes them out. Takes them out. If you reduce the number of CD4, guess what? There's no one there to regulate the other guy. They're helpless, right? They don't work. They don't work without this guy telling them what to do. Make sense? They're like soldiers without a general, and they don't know where to go. That's why people with HIV who eventually get AIDS, right, end up having all types of opportunistic diseases that you normally don't see. Because they have a non-functioning immune system. Why? The general is gone. The soldiers are lost. Make sense? Last one, hit it.
Helper 1 regulates the T cells.
TC and macrophages. TC and macrophages. The regulatory guys, okay, so TR regulatory, we won't spend much time on them because all they do, let me back up, they used to be known as the suppressor T cells. Get back up. So TR used to be known as suppressor T cells because they suppress autoimmune diseases. So autoimmune means what? Your T cells or even your B cells are attacking your own cells. You don't want that, right? So their job, their regulatory guys, are to suppress autoimmune disorders. So we won't spend much time on them, but we'll spend a whole lot of time on these guys. Make sense? Okey-doke. Now in this table here, you don't have to spend too much time on it, but simply put, you see I talked about the B cells, right? Great. And we talked about the two types of what? Helper T cells, right? There are two types. I said what? Type 1, type 2, right? Cytotoxic T cells, great, and regulatory. Let's get into what these guys do and how they tell the other guys what to do, right? Now, I mentioned to you in my last email, right, sometime yesterday, I said we got to skip some stuff for time's sake, but really, you don't have to know about that stuff, right? So if I put omit there, that means skip it. Don't even worry about it, okay? So skip that. All this stuff here, skip that, please. All this stuff here, put an X on it. Okay, here we go. Cytokines. Yeah. Cytokines. Now, basically, how do the immune cells, whether they be B cells or T cells, even macrophages, how do they communicate with each other? Okay, how do they talk to each other? They use cytokines, okay? So simply put, cytokines are soluble proteins that are used for intracellular communication. And when I use that prefix intra means within, right? Between, within. So this is intracellular signals. This is how your leukocytes talk to each other. Okay? Now, blah, 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 yeah, cytokine network, it's like, AT&T or, Verizon. It's basically how they talk to each other, okay? That's all that it is. Now, there are several different types of cytokines, and you should know what they are and what they do. For example, again, cytokines are intracellular communication signals, like little cell phones. This is how they being leukocytes, talk to each other, okay? you got interleukins, or IL. So interleukins, leukins come from leukocytes, right? These are signals among leukocytes, like B cells and T cells, okay? Interferons, we talked about them in the last chapter. This is not new. So interferons, we learned them as basically antiviral proteins, right? They protect other cells from getting the same virus that their neighbor died from, right? But in this case, interferon gamma, so I repeat, interferon gamma is secreted by Th1, so T helper cell 1, and it stimulates macrophages, okay? It makes them turn on, okay? Growth factors basically stimulate the stem cells to produce more leukocytes. You always have an ample supply of B cells or T cells. Okay. TNF, so tumor necrosis factors, secreted by either macrophages or T cells to literally kill tumor cells in the name itself. So take out cancerous cells, so TNFs. And the last ones are chemokines, okay? Chemokines are basically signals that attract your leukocytes to some location. Like, for example, if you want your leukocytes to go to an inflammatory site, you would produce chemokines to say, hey, come to this section over here. We need your help. Makes sense? So chemokines literally signal leukocytes to come to this area. We need backup, okay? Okay, now more about this MHC, this major histocompatibility complex. Everything up here is like real trivia, in my opinion. What you should know is the very last point. The very last thing I highlight is what you absolutely need to understand in terms of how they work, okay? Basically, what an MHC does, it literally presents epitopes. That is it. That's their job. They present an epitope. And some other cell will come around. Normally a T cell will come and examine it, and then it will be activated from there. So the only thing you need to know about MHCs is that they present epitopes. Okay, that's it. Real easy. Now, there are two, and yes, they have hands. In other words, you should know by now, if you recognize or bind an epitope, you should know they have hands, right? You should know that by now. So yes, they have their own little hand here to recognize and bind an epitope. Real quick, there are two types of MHCs, and yes, I'm going to tell you right now, you should know who has them, okay? Yes, you should know who has them. There are two types of MHCs, MHC1 and MHC2. Now, I'll tell you right now, when we get into how things work, in other words, who gets turned on, it's going to be who binds MHC1 versus MHC2 and blah, 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 okay? Real quick. Now, before I get into words, I put the slides out of order because I believe it's better to look at the picture before we get into the words, okay? So right now, we are looking at a cell membrane, and we see two types of MHCs, right? With their own what? Hands, right? They're going to bind something. Some what? Epitope. You should understand that every hand, right, only binds what? One epitope. But of course, you have thousands and thousands of different types of MHCs, right, on different cells. Got it? Okay. Now, you should understand that class 1 is found on every nucleated cell, with the exception of what?
Precursors.
So red blood cells, okay? Red blood cells lose their nuclei. So all cells, right, all cells except for RBCs, they have MHC. In other words, all cells have the ability to present an epitope, right? That's all they're doing. Hey, look at this thing. It doesn't belong in our body. You should examine this. That's what they're doing, right? Look at this. Look at this epitope. So all cells, with the exception of what RBCs, they can potentially present an epitope. Okay. Now, class 2 are found on what we consider our immune cells, okay? So the only way you get both types, because all cells have class 1, right? Except what? RBCs, all right? Now, who gets class 2? Now, keep in mind, if you have class 2, by default, you have what? Class 1, right? So if you have class 2, again, by default, you have class 1. Now, if you have class 2, you are really an immune cell, right? Your job is really for that function. So think of macrophages, right? Macrophages would have both, right? I mean, these are immune cells. So immune cells would have both types. They say B cells, right? Aren't B cells immune cells? So immune cells would have both, okay? Dendrites, so dendritic cells, I talked about them last chapter, they have them too, okay? So keep that in mind. Ooh, new acronym. I like this chapter. It's like the government, right? All these acronyms. APC. An APC, you should know, it's an antigen-presenting cell. So B cells, macrophages, dendritic cells, these are basically immune cells, and their primary job is to present antigens. So they're called antigen-presenting cells, or APC for short. So whenever you see APC from here on out, right, it's what? Antigen-presenting cell, right? Main job, look at my hand, right? Look at my hand. That's what they're doing. Ooh, more pictures. I mentioned these guys, these dendritic cells in the last chapter, right? They're basically found in your skin, also in the mucous membranes, and they're kind of looking around for things that don't belong. They have these long extensions called dendrites, and they're sampling what's out there. That's what they're doing. They're sampling what's out there. They touch something, right? This don't belong, right? They kind of process the antigens, right? So that they can do this, right? They do that. And these guys, we'll talk more about them because these guys play a major role in terms of letting T cells know something doesn't belong in the body. Makes sense? So they're dendritic cells. They are a major part of the APCs, right? Antigen-presenting cells. That's their primary role. Blah, blah, blah. We're not going to spend time on antigen processing because all that means is some cell, some dendritic cell, sees some foreign antigen with an epitope, it's going to sample it, and then basically do what? Present it, okay? You don't have to understand how it does it. Just understand that when these APCs find antigens that don't belong, they do what? Present them on the outside so that T cells see it, right? And then we get the whole process started. This is what specific immunity, right? It's not turned on all the time. That's going to be bad. We got to control it, right? Control those TC guys. You don't want them running rogue, right? That's dangerous. Control them. So that's why I said omit, right? Don't worry about how they're presented. Just know that they're what? They are presented on what? MHCs. Make sense? Know that part. So omit all that. Now, at this point, we will get into the nitty-gritty of cell-mediated immunity and also humoral immunity. First, that will be what? Cell-mediated immunity, which means we're dealing with what? T cells. Got it? Activate T cells. So we already know everything on there, right? That's all review, correct? Don't we know all that stuff? Absolutely. So let's keep going. It's basically T cell-mediated immunity. We know that part. Here we go. How do you actually turn on the T cells so that they work for you? Remember, this is specific, right? They should be only turned on when you need them. Okay? You've got really 4 basic steps. And keep in mind, let me just back up. If you're talking about cell-mediated immunity, what you want to do is activate the killers, right? Activate the TC cells. How do you turn them on so they take the bad guys out? So that's what we're talking about. If you know nothing else, if I say cell-mediated immunity, we're talking about how do you turn on the TC cells, correct? Now to do that, you've got 4 steps. And yes, we'll go into all 4. Now I said in the study guide, you should know the basics, okay? Because in the textbook, they go into interleukin 12, interleukin 2, okay? I'm not going to go into that much detail. But what I am going to present to you is what you should know. Make sense? Okay. Real quick. And if I put new, that means it's new. Don't worry about it. If you want, like Faye's been doing, Faye's like, I'm not writing that, I'm taking a picture. You can do that. I can upload this Angel this afternoon, or like a good student, just write it down, okay? Just write it down. It's not that bad. Writing it hadn't killed anybody. Back in my day, I'm telling you the truth, back in my day, we didn't have Angel. You just showed up to class and you wrote everything you saw, okay? And I'm still living. All right. First step, okay? Remember, activate TC cells, right? Activate your killers. First step is what? Antigen presentation, okay? So basically, simply put, you have some, for example, a dendritic cell. Remember, they're looking around, they're sampling what's out there, they find some antigen that does not belong, and they're going to present it. They're going to present it, back up, okay, they're going to present it on their MHC. Now remember I said these guys have both, right? They have both MH2 and MHC1, okay? So for this process, they start off with what? MHC1. Basically, hey, look guys, something is around here. You should take a look. That's what they're doing. Now what they do is they leave the mucous membranes, or they leave your skin, and they literally travel to the lymph node. Literally. It takes them a few days to do that, okay? It's not instantaneous. They have to move, okay? They literally travel to the lymph node because that's where some of the T cells are hanging out, okay? And when they get to the lymph node, they're like, hey guys, sample this. Some TC, right, with its appropriate TCR will bind that guy's MHC. Now, of course, there's a picture of this, right? But I figure present the words first, and then I'll show you the picture. So in other words, what's happening is you have a TC, right? Because we want to turn him on, potentially. And guess what? He has a TCR, correct? And I said a few slides back, didn't I say that T cells do not directly bind epitopes? They have to do what? They have to bind an epitope that's complex with what? An MHC, okay? So we've got a dendritic cell, for example. It can be any APC, right? He presents his epitope on his MHC1. This guy sees it, right? And with his TCR, he binds it, right? That's the same MHC1. They've got the same one, right? They hook up, literally, they hook up. But that is not enough to fully turn him being TC on. So even though they've literally done that, that's what they've done, they're touching, literally, they are touching. That is not enough to turn on that TC cell. It's not enough. Okay. Next step, helper T cell differentiation. In other words, how do we activate the TH cell so that we fully activate the TC cell? Remember, a couple slides back, I said that T helper cells, right, there are two types, correct? Didn't I say that? I said the TH1 guys do what? Activate the TC. I said that, right? No, I did, okay? Great. So, next step. This is already happened. Great. Step 2. We've got to get him turned on. So, the T helper cell, he comes along, right? They're all in lymph node, okay? He comes along. Now, he is going to bind the MHC2 on the same APC. So this guy right here, remember, didn't I say APCs have both types, right? If they are an immune cell, they're going to have both types, right? MHC1 and MHC2. Now keep in mind, they're going to be binding some epitope, doing the same epitope. So far, so good? You got to know this, I'm telling you straight up. All right. Now, this helper guy, he binds the MH2 on the same APC, this guy way up here who started the whole thing. And at that point, that T helper cell is now going to become a T helper 1 cell. You're like, why should I care? Because the T helper 1 guy is now able to turn on the TC. Got it? Remember, what did I say? I said if you take out these guys, you can have all of this in the world, right? You can have TC binding to this guy, but if he's not turned on, nothing happens. Okay? Nothing happens. That's HIV. And that's why they die eventually, right? Yeah. T helper guys turn the other guys on. Okay? So at this point, when he becomes TH helper 1 or TH1 for short, he's activated, right? He's turned on, and basically, all he's going to do is what I just told you. The only thing he's going to do is literally tell him to now turn on. He's going to activate the TC guy. That's it. He activates TC. And at that point, when the TC is activated, he's going to start to do what? Making copies of himself? Is that a hand?
Yeah.
Hit it.
In the very beginning, you said TH activates the...
Which one? I said there's two types.
TC.
Which one? There are two types of THs, right? I said TH1 and TH2. Which one?
TH1.
TH1. Okay, so when TC is activated, now how did he become activated? I'll tell you right now, he got a signal from who? TH helper 1. You got to say 1, right? At this point, TH helper 1 is going to activate who? TC. So TC literally gets the signal from who? TH1 to say, hey, turn on, make copies of yourself, get activated, we got a battle going on. And at that point, keep in mind, at the point TC is activated, he is now ready to attack. I repeat, at the point that TC receives a signal from who? I keep trying to repeat it, right? Repetition is key, right? Yeah. When TC gets the signal from TH helper 1, it's now turned on. At that point, he's ready to attack. He's going to make copies of himself, but at that point, he's ready to attack the cells that are basically causing the problem, right? So activation means two things: kill the bad cells and make more of yourself. Okay, that's what it means to be activated. Now, some of them, so a small percentage, so most of them, when they proliferate, they're going to go out to battle, okay? They're going to go out and fight. But some of them stay behind, and they eventually become your memory T cells. So I believe somewhere in my study guide, I said you should understand where these memory cells come from, okay? So whether it's a memory T cell or a memory B cell, they come from when these cells are activated, right? And they start to replicate themselves. Some of them go out to battle, but some of them stay behind and just become your memory T or memory B cell, okay? The last part here is self-stimulation. So at this point, the TC cells no longer need two signals, right? It was two signals, right? The APC guy and the TH1 guy. Isn't that two signals? So at that point, by the time you've done all this, you've reached the last step. That means they no longer, they being TC, they no longer need signals from APCs or TH1s. They can go on and proliferate. Ooh, proliferate, that's a fun word to say.
Replicate.
Yeah, replicate, there you go. Basically, they can replicate without any more help. Make sense? Because to get it all started, they needed two signals, right? That makes sense? To get them going. But by the time they get here, they don't need help. They kind of stimulate their own cells to do what they need to do. Make sense? And at that point, they go out and they fight. So basically, they find the cells that are presenting those epitopes. Again, these are cells that are affected with the virus, right? Remember, intracellular parasites. So they find those cells that are presenting, hey, help me, there's a virus in me. And they basically cause those cells to kill themselves. So simply put, you should basically understand that TC cells cause the infected cells to do what? Apoptosis. Remember I introduced that term on Thursday? That's what they do. They cause the infected cells to kill themselves. That is it, okay? That's all you should know. That's how they kill other cells. Hit it.
Okay, so to start the whole system, you have the APC cell recognizing an epitope.
Mm-hmm.
Okay. From what I understand, we have the receptors for all the epitopes. What happens if the system
Not technically.
Okay. Well,
The MHCs? MHCs?
Well, what I'm saying is that, like, what happens if there's a new virus comes along and it has a different set of epitopes that we have never encountered before, and we have no...
Yeah, that's true. Because you only have about 100,000 different MHCs, which is
Right.
not a lot.
Right.
So you may not have the right MHC to begin with. So in other words, to understand that concept, some people, for example, have allergies, some don't, right? Like I have hay fever. Somebody may not. It's because I have MHCs that bind pollen and tell the T cells, hey, this is a bad additive, but it's basically pollen, and I get sick. But if you don't have that MHC, right, this makes sense? If you don't have that MHC that binds pollen, guess what? You will never have allergies. You never have to take Claritin or Nasonex or Zyrtec or any of the other ones. I tried them all. Makes sense? So you really don't have all MHCs.
Right.
You don't.
But what happens if something really bad comes along, and now can you just not even fight it?
That's
No.
right.
You develop memory.
After the
But
first
you
attack.
need
Right.
You
the
need
MHCs
to
to
make the
stimulate
memory
that, because we'll get into that. Without MHCs,
Right.
you never get to memory cells.
Right.
You never ever get there.
I mean, that's my question. Is there a way you can acquire more MACs? Is there a process
The
for
best
that?
thing you can do is you can, how would you do that?
Take
Right.
home ones?
You can't make,
Craig?
you can make antibodies through a T cell independent pathway, but that's the best thing you can do. You can make antibodies to kind of take care of that particular pathogen,
Right.
but it has to be a T cell independent pathway, which we'll get into today.
Okay.
What if you're allergic to MACs?
Same difference.
So your MACs are
Same difference. Look at it. Blah, blah, blah. Okay, great. Now, if you're looking at the picture in the textbook and you like pictures, I'm going to walk through this real quick because we got to finish chapter in like 30 minutes, okay? Plenty of time. Here's step one, right? This guy looks like a dendritic cell, right? Remember I said he's an APC. He goes around looking for antigens, right?